Hello, everyone. So, welcome to Chapter 9 on human development. So, this chapter can be a big, challenging one because it's going to cover human development right from the moment of conception all the way to old age and death. So, we're going to start with defining what human development is, how do we research it, what are some ongoing debates in development, and then we're going to break up the lifespan into different sections and talk specifically about all the things that go on. So we're going to start even prenatally. Um, you're going to learn a little bit about genetics and what happens even before you are born. Then we're going to cover infancy and childhood and talk about physical development, cognitive development, psychosocial development, and then pretty much similar thing in adolescence, physical, cognitive, psychosocial. We're going to start talking about moral development. And then adulthood, we're going to address the same kind of things going on in adulthood. So let's start at the beginning with defining what development is and talking about what are some of the ongoing discussions or debates in development. So developmental psychology is the branch of psychology that studies how we change over our lifespan. So studying how we change physically, cognitively, socially, and emotionally. And we're going to see that there are three main debates in developmental psychology. So the first debate we're going to talk about is sometimes referred to as the stages or continuity debate. As we go through this chapter, we're going to see that how some theorists talk about how we change physically, cognitively, socially, and emotionally through the lifespan, they do so in a different way. Some of the theorists describe the lifespan as a gradual, ongoing, consistent change process over time. Whereas other theorists talk about stages. They break our life up into a particular age group and then talk about how something big and important happens at that stage. And then once we accomplish that, it sort of marks the move on to the next stage of development. So that's something people disagree on. Some see development as a continuous gradual process of changing, whereas others see it as sort of this stop-start kind of development in stages. Probably the biggest debate is known as the nature versus nurture debate. So this is the debate that's basically saying what has the biggest impact on how we change cognitively, physically, socially, and emotionally. Are the influences on development things that you have no control over, like your genes? That is what is the nature side of the debate. Or is the bigger influence coming from the nurture side, which refers to things outside the person, how they are brought up, the parenting that they've had, the education that they've had, all of those kind of things. And then the third ongoing debate is stability and change. There's disagreement amongst some developmental psychologists about where should we focus our interest in our research. Should we focus it on those things about us that remain pretty consistent, or should we focus on the changes that happen over time? As we get into some of these topics, we're going to see that even though on the surface we might seem we change dramatically from the time we're little kids all the way to adults, when we get to things like temperament, and I'll give you all sort of an informal assignment to do to, if you got your parents around still in your lives, talk to them about what you were like as babies. When we talk about things like temperament, we see we don't change a whole lot from when we were little kids. If your mom says you were a easily upset kid who cried easily, that's probably how you are as an adult as well. So there are some things about us that do stay pretty remarkably consistent. So those are some things that people disagree on. So now we're going to talk about how research is done in developmental psychology. There's three main research designs that are used. So we're going to talk about cross-sectional studies, longitudinal studies, and then cross-sequential studies. In a cross-sectional study, what we do is we choose some area of development that we're interested in. We pick different groups of people of different ages, and at the same time in history, we are gathering data on them and comparing the different groups. In longitudinal studies, we're starting with one group and we're staying with that group and following that group and gathering data from that same group of people at different points in time. And then cross-sequential combines elements of both of those approaches. So let's look at what these are like individually and the strengths and weaknesses of each of them. So in the cross-sectional studies, it would look like you see in this infographic that you have in your textbook as well. So we've got groups of people, 3 different ages. At one point in time, we're gathering data on those 3 groups of people, and we're comparing them to one another. It can be a very quick and efficient way of gathering data on people of different ages, but it doesn't always take into account that those different groups of people of different ages have experienced different histories. They are susceptible to what is known as the cohort effect. A cohort simply refers to the age group that you belong to. So let's say, for example, we did a study where at one point in time, we took people in their early 20s, like probably most of you, and people in their early 50s, and we compared them on one developmental aspect, let's say intelligence. And we found a difference. Let's say that the 20-somethings scored higher than the 50-somethings. Does that automatically mean there's something inherently different about those groups that is related to their cognitive ability? What else could account for those differences that a comparison like that at one point in time doesn't take into consideration? So think about that for a second. What would be different comparing someone like me to someone like you? It could be something like the presence of technology. For someone who's in their early 20s, you have had computers, handheld devices since the time you were born. Whereas someone my age, I literally did not see a computer until I was in 9th grade, and it was nothing like the computers that we have now. So there could be other things that could account for the differences that have really nothing to do with development. Now, a way of taking care of that problem is to do longitudinal research. So in longitudinal research, we start with one group of people and we follow that same group. So every 10 years in this example, we find the same people, gather data on them, and we talk about changes over time. They're all of the same cohort, so that means they've had the same kind of histories, the same kind of advances in technology, the same kinds of educational advances. So it's addressing all of those cohort effects. However, it can be a very costly and long process to get the kind of data you're really interested in. And there are some well-funded longitudinal studies that have been going on for decades. They started when people were infants, and now these people are parents and grandparents. So what kind of problem might you see with a longitudinal study besides the time and cost it takes? What do you think might happen to my sample when I try to get that same group every 10 years to gather data from them? So if you're thinking they might die, they might lose touch with the researcher, you're thinking the right way. So a big problem with longitudinal studies is as they go on, your sample size tends to get smaller. And if you take your minds back to that second chapter where we covered research methods, we know if we got small sample sizes, it's harder for us to draw some conclusions and make generalizations. So that's the biggest problem with longitudinal research. Now, in cross-sequential research, it is sort of taking the best of both worlds. We're sort of seeing a combination of both cross-sectional and longitudinal, so that we are comparing different groups, sort of like a cross-sectional, but then we are covering the same groups at different points in time. And if this were to go on, we'd always be adding, a new cohort the next time, and it's constantly replenishing your population. So it's addressing those cohort effects and showing changes both within the individuals and within the groups. Again, this can be a very costly and time-intensive way to gather data, but it's a very thorough way of addressing the shortcomings of both cross-sectional and longitudinal research. All right, so the next objective we're going to talk about is you're going to learn a little bit about genes and how they influence development, and particularly how they determine our biological sex. So in every cell of your body, except for red blood cells, you have a nucleus. And within that nucleus, you have what it takes to build a whole new person. Within the nucleus of every cell that has a nucleus, you have 46 chromosomes, 23 pairs. You get 23 chromosomes from your biological mother, you get 23 from your biological father. Each chromosome contains a molecule of DNA, deoxyribonucleic acid. And it is the DNA which encodes for what it takes to make a new you. Now, when we are just learning genetics in a very cursory sense, like we are in an intro class, we might talk a little too simplistically about genes, and you will hear, people say, well, there's a gene for this trait and a gene for this disease and things like that. That really is oversimplifying things. It's a very, very complex process, and all genes really do is tell amino acids how to assemble into proteins. Now, it just so happens that we're all made of proteins, but there's quite a lot that happens between a gene encoding for a particular protein and you manifesting an illness or a disease or something else. It could be that protein is used to make a particular neurotransmitter. And if you don't have that protein, then you're not going to have the right amount of that neurotransmitter. And if you don't have the right amount of that neurotransmitter, it could manifest in the symptoms of major depressive disorder or something, along those lines. So all of your cells have those 46 chromosomes. The exception are your sex cells. So the sperm in the male, the egg in the female have just half of the complement of chromosomes. They have 23. So that when a mother and father have a new child, you get that mixing of the unique genetic material. The 23rd pair of chromosomes is the one that determines our biological sex. So the sex chromosomes are characterized by the letters X and Y. So if you have two X chromosomes as your 23rd pair, that is the genetic formula for a female. If you have an X and a Y, that is the genetic formula for a male. Now the next concept to know is what's the difference between a genotype and a phenotype? So you have two versions of every gene because they're coming in pairs, one from your father and one from your mother. Sometimes what those genes are coding for, they're sending the same kind of message. Sometimes they are providing conflicting messages. This is what we refer to as dominant and recessive genes. Dominant, like the name suggests, no matter what it's paired with, it's going to dominate. Whatever it is encoding for, those instructions are going to be followed. A recessive gene is only going to show itself, it's only going to be expressed if it is paired with another recessive gene. So as an example, your ability to fold your tongue over like that is the result of a dominant gene. So if you are unable to do that, that means you have inherited both recessive genes from your parents. So this is a schematic of how the genes for this trait could distribute if these two individuals got together and had children. So each of them are carrying one dominant trait and one recessive trait. So this kid here is getting the dominant trait from both the mother and the father. So they would have a 25% chance of having a child who had two dominant genes. Because both parents are carriers of the recessive gene, they would have a 50% chance of having kids who carry the recessive gene. Neither of these kids show the trait because they have the dominant gene and it is overriding. They would have a 25% chance of having a child who gets the recessive from both the mother and the father. This is the only kid who would show the trait. This is the only child who would not be able to roll their tongue. These two would be carriers. They're not going to show the trait, but they could pass it along to their own children. So this is just a table of some common human traits that are governed by dominant and recessive genes. So, an easy one to think of is if you are naturally blonde-haired and blue-eyed, that means that you have the recessive gene for those traits from both of your parents. So that means either both your parents were blonde-haired and blue-eyed themselves, or that they both were carriers of the gene. So if you have two brown-haired parents and you come out, blonde, doesn't mean mom had an affair with the mailman or something. It could just mean that both of your brown-haired parents carried the recessive blonde-haired gene, and they had a 25% chance of having a kid with blonde hair, and you are it. So the 23 pairs of chromosomes that you get at the time of your conception is what is known as your genotype. It is what it is from the moment you were conceived. It does not change. However, your genotype does interact with the environment. The environment can influence how those genes show themselves, and there are things in the environment which could turn on the expression of that gene or turn off that expression of the gene. So the outcome of your genes interacting with the environment and what we see as the end product is what's referred to as the phenotype. That whole study of how genes and environment interact is known as epigenetics. So let's take, for example, a trait observable like skin color and texture. If you have identical twins, they have identical genotypes. That would mean the genes that they have that are providing information about skin color and texture are the same. Does that mean as they get to be adults that these two people are still going to look exactly identical in terms of their skin color and texture? Not necessarily. You might have one of the twins who smokes, spends a lot of time in the sun and in the elements. Those are going to have an impact on their skin. So what we see in the end in terms of the phenotype might actually be different, even though we started out with the exact same genetic material. All right, so now we're going to start with prenatal development, starting at the moment of conception. Conception refers to the moment when a single sperm penetrates the cell wall of a single egg and fuse, joining that unique combination of genetic material. So in this section, you have a lot of opportunities to delve into these topics a little more thoroughly and giving you sometimes animations down at the level of the cell to better understand it. So one of those opportunities is through this active psych demonstration here. So I encourage you, you can pause me, check that out, and then come back. All right. Likewise, you have a link here to a video about prenatal development. I encourage you, again, pause me, take a look at that video, come back and be sure that you understand all of this. What you really need to take away about prenatal development is that there are 3 stages to it. And you need to know what those 3 stages are and what are some of the big things that happen in each of those stages. So from the moment of conception, when the moment that egg and sperm fuse and join their genetic material, marks the beginning of what's known as the germinal period. And this goes on for approximately 2 weeks. So at this stage, the developing human being is just a cluster of rapidly multiplying cells that we call a zygote. And in those 2 weeks, that rapidly developing cluster of cells moves from where it was fertilized, usually, the mother's fallopian tubes, to the point where it is going to attach to the mother's uterus. When that cluster of cells attaches to the uterus, then that begins what we call the embryonic stage. And we refer to the developing human as an embryo now. So once that cluster of cells latches onto the mother's uterus, we see an incredible amount of stuff happen in just the next 6 weeks. We see 95% of your organs and tissues formed already. So by the end of 8 weeks after conception, even though you might still be super tiny, you will have a beating heart, recognizable organs and limbs and body parts. Then the longest part of prenatal development goes from 9 weeks to the typical delivery, which is about 40 weeks, the fetal period, and we call the developing human the fetus. So the groundwork for everything was laid in the embryonic stage. The fetal period is a time of maturation. So all the groundwork that was laid gets matured, all the body systems mature, everything gets bigger. Now, for humans, something that really is of interest to us in development is the development of the brain. So again, you have an active psych demonstration link there that will take you down to the level of the cell so you can see some informative animations about what is going on for brain development. So if you look at those animations, you will see that we begin the embryonic stage as just like this 3-layered cell that ultimately folds in on itself, and it's going to form what's known as a neural tube. One end of that neural tube is going to bulge out and ultimately form the brain. The other end is going to end up forming the spinal cord. Four months into the pregnancy, the brain is developed enough that it is starting to send neural impulses to the muscles. Around 4 months into the pregnancy is when the mother can typically start feeling the baby kick or move within her. That's known as quickening. Because you have a rudimentary brain already at 4 months into the pregnancy that's sending neural messages, means that your senses are already becoming active. So the baby can hear things in the prenatal environment, hear the mother's voice, can hear the bodily noises and things like that. 6 months, we have a lot of growth in the brain that if you paid attention to the video and they were interviewing the developmental psychologist, one thing that she said that really strikes me is that for every minute of prenatal development, you are making 250,000 neurons. So in that fetal period, every minute, you're making 250,000 neurons in the brain. During that last trimester, months 6 through 9, everything finishes up. You start myelinating your neurons. We're going to see brain development's going to continue throughout the life, but so much has happened during that fetal period that again, 250,000 every minute. That's just like really impressive to me. So here is a schematic that you have in your textbook as well that just sort of breaks down the timelines for the development of the different parts of your body. Now, as was stated in the video about prenatal development, the placenta is a very unique organ, only present in a woman's body when she is pregnant. And it is an organ which transfers nutrients and oxygen from the mother to the fetus. A teratogen is the term that is used for anything that could get transferred to the developing baby that could harm development. So that could include things like viruses, it could include things from the environment, it could include things that the mother is ingesting, like cigarette smoke, alcohol, things like that. Some teratogens have been well studied and well established, and cigarette smoke and alcohol are the most common of them. High levels of alcohol during pregnancy results in a very specific set of symptoms known as fetal alcohol syndrome. The baby will be born with subtle, disproportionate malformations. They will simply look funny and different. They will have smaller heads, different facial features, and that alcohol will have affected the brain development, which can result in cognitive problems throughout the child's life. Now, the effects of teratogens are most prominent during the first 12 weeks of pregnancy, and many women, particularly if it's a woman who might have had irregular periods and things like that to begin with, might not even know they are pregnant during those first 12 weeks. So if they were smoking and drinking even, moderately during then and would have stopped if they knew they were pregnant, they might still damage the unborn child's development simply because they're not even aware that they should have altered their behavior. So you have this chart in your book. I encourage you to check this out. These are just some well-known teratogens, and the effects that they can have on the developing fetus. All right, so now let's get born. Let's talk about um, physical changes that happen in infancy. So we come into this world right from birth, already able to do some stuff. We are born with some reflexes. You have a link to a video about newborn reflexes. I encourage you to pause me and take a look at that video. These are things that doctors and nurses will check out immediately after birth to evaluate the infant. The presence of these reflexes are a good sign. The absence of them might mean, something developmentally is off. Maybe we have a premature baby. So some of the common reflexes are things like the rooting reflex. So if you stroke a newborn baby's cheek, they will automatically turn their head to that direction and open their mouths. That's something they do automatically. It helps them find their mother's breast so that they can start nursing. You put anything in a newborn baby's mouth, even a finger, and it automatically starts the sucking reflex. There are some very interesting reflexes, like if you stroke a baby's feet, their toes will sort of curl over. If you surprise or act like you're going to drop a baby, they automatically fling their arms out and bring them back to the center. Evolutionary psychologists suggest that those are reflexes that are probably vestiges from our tree-dwelling, more ape-like ancestors, that if you were a baby monkey falling out of a tree, it would make sense to try to grab onto something to break your fall. So we come into the world right from the moment of birth, able to do some of these things automatically. Some of those reflexes go away. Some of those reflexes get replaced by voluntary behaviors. So the sucking reflex is an ideal one. You can suck immediately from birth, but as the baby starts getting accustomed to being fed, whether by breast or bottle, they start gaining more control over their sucking behavior. And if you have had a baby, you will know that as they progress through infancy, it gets easier and quicker to feed them because now they're no longer sucking by reflex. They're, willfully controlling it. So this is just an example of some common infant reflexes that you will see in that video. In terms of sensory capabilities, their visual acuity and their ability to focus on things visually is best at about the distance that someone would hold a baby, so about a foot away. And they assess what kind of visual stimuli hold a baby's attention by simply timing their gaze. So showing them different things and seeing what are they more paying attention to. And if you think of how baby toys are designed and how we decorate nurseries, we take these kind of things into consideration. So they're drawn by concentric circles, by contrasts, by sharp angles, by bright colors. These are things that will attract and hold a baby's visual attention. Now, the visual system is going to develop and continue to do so over time. In our sensation and perception chapter, you will recall that one of the things we are able to do with two eyes on the front of our heads is to use that to judge depth and distance. That is something that takes a little while to develop. And one way that it can be assessed whether a child has developed that yet has been this visual cliff design. So you put the child in this apparatus where here you have a clear plexiglass floor. So a child could crawl out on here and not fall through. A child who has not yet developed the ability to judge depth and distance with binocular vision will crawl out onto that floor because they have not yet been able to judge that it looks like it's going to fall. A kid who has developed that depth and distance, when they get to the edge here, they'll stop themselves because they will, perceive that it is a cliff and that they're going to fall off. Usually takes about 6 months into infancy to develop that kind of depth perception. In terms of our other senses, very soon after birth, a child, again, if you're in with that distance of holding the baby about a foot away, a newborn is already able to do some basic imitation, which is essentially for forming a bond, for learning how to socialize. So you smile at them, they will smile back. If you stick their tongue out, they'll stick their tongue back. I just saw a cute little online video of a father changing an infant and doing this. So the dad would start dancing in front of the baby, and when the dad would dance, the baby would be all excited and shake their hands, and then the dad would suddenly stop and stay still, and you would see the baby do the same kind of thing. They're able to recognize their parents' voices very soon after birth, particularly the mother, because they have been hearing the mother's voice even prenatally, although it's somewhat distorted by, passing through the abdomen and such and the bodily fluids, but they're able to recognize their parents' voices within a day or two. Smell as well. Babies are able to detect the smell of their own mother's breast milk, for example. They respond to touch. Touch is very important, particularly for stimulating the development of newborn or preemies in particular. So baby massage is a real thing. They need touch. They need, rocking. Kangarooing is something that is commonly done with babies who are born a little premature. Like the name suggests, you carry the baby around like a kangaroo. Kangaroos carry babies in pouches. In kangarooing, you literally strap the baby on to the mother so that they're constantly having that skin-on-skin contact, and that facilitates the development of the child. So we're going to see that although we had some tremendous amounts of brain development occurring even prenatally, brain maturation is something that's going to continue going through adolescence and beyond. And that the more mature and branched the neural networks become, that means the more synaptic connections they can make. And as the brain matures, so do the things that the baby is able to do in terms of walking, talking. And we're going to see that throughout the school years, we see that brain maturation continue as you learn more and more complex things with reading and writing and math. So we have this tremendous growth of neurons. we end up with sometimes more connections than we need. So when we get into adolescence, we're going to go through a process known as synaptic pruning. So you know what pruning means when it comes to a tree. You're cutting off the wild growing branches or the dead branches. Pretty much means the same kind of thing in adolescence, that synapses are pruned. Ones that are superfluous or duplicated or not efficient are cut away, so that by the time you're an adolescent, you're left with a more and more efficient brain that's able to send very efficient neural messages. So things that you're not using or that are redundant or confusing, stop, so that you have just the most efficient brain you can have. Rosenweig is a classic study showing the impact of exposure from the environment on brain development. This is what we call an analogy study. It was done with rats because to do the same thing on humans would obviously be unethical. So we do it with lab rats, and then we try to draw an analogy to how it would play out in a human brain. So Rosenweig took 3 groups of lab rats and raised them in 3 different environments since birth. One was raised in what he termed a rich environment. By that, he meant the rats had a lot of things in their environment to stimulate them, things to explore, things to investigate, things to play with, other rats to socialize with. That is a rich environment. Some rats were just raised in a standard rat colony with other rats in normal cages like rats typically were raised, with nothing special to play with or to investigate. And then finally, the third group of rats were raised in isolation. No other rats to socialize with, no things to play with, nothing to explore or investigate. And then at the end of their lives, those rats were autopsied, their brains were investigated and weighed, and they found that the rats that were raised in the rich environments had the heaviest brains, meaning their brains were most developed. Under autopsy, they had the most synaptic connections. The rats raised in the poor environments had the lightest brains with the fewest synaptic connections. So again, if we were to draw analogies to human beings, then that would mean it's very important to stimulate a baby and child's brain through giving them a rich environment where people are interacting with them, reading with them, exposing them to things, giving them things to play with and investigate. It actually has an impact on brain development. Now, obviously, a big thing that happens in infancy is language acquisition. So we talked about language just last chapter in cognition, language, and intelligence. So how do we acquire language? We sort of addressed some of it last chapter, and we said there's people who advocate for different things. One approach is the behaviorist approach, saying we learn language because we associate words with what they represent in the environment. Speaking is reinforcing. We are learning through observation and modeling. Parents and teachers and other adults will praise us when we speak and correct the things that we say wrong, and that is a big way of how we learn. You'll also remember, though, that from the cognitive perspective, folks like Noam Chomsky said there's got to be something more. And again, that's what he called the language acquisition device. He said the fact that kids all over the world learn language at approximately the same pace and the same sequence and so quickly, and that you are able to generalize rules of grammar without having to hear an example of everything you will ultimately say in your life, tells us that there must be something in our brain that sort of we're born with that prepares us to learn language so quickly. If, again, you remember, we emphasized the interactionist approach last chapter, that we really do need both. Even if we have a language acquisition device, a LAD, you still need exposure to language to learn your language. And studies in infant development related to language tell us that there's a particular way of speaking to babies that facilitates language development. We call this infant-directed speech. Now, you put many adults in the presence of a baby, and pretty much everybody does this almost naturally, probably not even realizing they're doing something helpful and that is research supported. Even, big tough dads will talk in a high-pitched voice. They will repeat things, sort of talk in that sing-song voice. That's what we refer to as infant-directed speech. And it's a good thing that most adults sort of do that naturally, because infants pay more attention to it when people talk to them that way. We don't talk to infants like little adults, and it's good we don't, because they wouldn't pay attention as well, and if they don't pay attention, then they can't learn language. Studies like this are really bringing what Rosenweig suggested with his rats into the world of humans and looking at how environment can affect development, particularly with speech. A lot of these topics relate. So last chapter we also addressed the most empirically validated theory of intelligence, CHC theory, and we talked about the different broad abilities. And if you remember, one of the things we said is a broad ability that's important for every type of learning at every age is comprehension knowledge, your accumulation of facts and vocabulary. Because so much of learning is verbal. If you have a context to put it in, if you can better understand words, your learning is going to be better. Therefore, talking and reading and exposing kids to language is helpful, even when they're toddlers. So what these researchers did is they went into the homes of people of different income levels and measured, on average, how many words per minute do the adults speak to or expose the toddler to. And they found that in toddlerhood, parents who were of high income, on average, exposed their kids to 35 words per minute. Middle income parents expose their kids to 20 words per minute, whereas low income parents expose their kids to 10 words per minute. Now, there is a lot of things that could account for that. It could be the conflicting demands on parents. If you are a poorer parent and you're worried about simply getting food on the table, you might be working hard, have less time on average, less energy to spend with your toddler. But that has an impact because there's a huge growth in vocabulary from toddlerhood to the point where a kid is starting school at age 5 in kindergarten. So if we're seeing these differences before a kid even sets foot in a school building, that's telling us that there are some kids, through no fault of their own, are already starting school at a disadvantage compared to their wealthier same age peers, who again, through nothing special they did, are starting school with an advantage. A larger vocabulary, part of that comprehension knowledge, which again is going to help you in every subject area at every age. So this is just that data graphically. Now, it is important that everybody have an appreciation for the scope and sequence of language development. There are some variability here, but these are time frames for most kids. So already by 3 months old, kids are making sounds. They're cooing, making the vowel sounds in particular. By the time a kid is 6 months old, they should be babbling. They should be combining phonemes, like saying dada, mama. They're not really words yet, even though sometimes they approximate words, and it is very common for a baby to say dada before mama, and us dads like to think, see, it means they like us better. It really doesn't. It simply means the phoneme d is easier to say than the phoneme m. But let us dads have, our delusions. By the time a kid is 1 year old, they should be saying their first real intentional word. And then about a year and a half, they are engaging what we call telegraphic speech. They're putting words together in phrases that are not full sentences, but they are conveying meaning. And from 2 to 3, those toddler years, the years when that study on language exposure was done, that is when we have this huge growth in vocabulary. Kids are learning lots and lots of new words. And obviously, again, the more they're talked to, the more they're read to, the larger that vocabulary will be. Such that by the time a kid is entering school at age 5, they are fluent in their native language. They will have a vocabulary on average of about 10,000 words. They can speak in full grammatically correct sentences. They can talk about things in different tenses. Again, vocabulary, part of that comprehension knowledge or crystallized intelligence, is going to be something that continues to grow throughout adulthood, again, dependent on how much you read, how much culture you expose yourself to, and things like that. It's also important for you to have an appreciation of motor milestones as well. Again, there's quite a bit of variability among kids in terms of the time frames, but they always follow a particular sequence. And if a kid is too far off the typical time frame, then you should be considered, you should be a little concerned, maybe seek an evaluation or something. One thing to keep in mind about how motor development typically progresses is it sort of goes in a direction of inside out. So that means that we are best able to control the center parts of our bodies first, and then it progresses out to our limbs. So that you first gain control over the center part, your trunk, and you're able to hold yourself up with your core, roll yourself over, sit up, and then the development moves out to control of the limbs, the arms and the legs, and the baby's able to scoot and then crawl, and then stand by holding on to furniture, and then walk. And then the last of the motor skills to develop are the fingers, being able to control fine motor control. So this is why, like in kindergarten, you have kids using those fat pencils and fat crayons and things like that. That's because the control of the fine motor skills in the fingers and hands is the last to develop. So development of control of your muscle progresses from that center outward fashion.